Good evening, everyone, and my name is Nandika. I'm a PhD student at IIC Bangalore, and welcome to a problem-solving session for week 10 of our experimental biology. Um, Today we'll be discussing problems relating to the content released this week, which is mainly discussing the beginning of PCR. So, as we know, PCR stands for polymerase chain reaction, which is a way of replicating basically DNA replication, as we know. So first let's discuss a little bit about how DNA replicates in our body. So as we all know, DNA is DNA is what stores the genetic information in our body. It's genetic code, and it is made up of repeating units of nucleotides. Okay, nucleotides have these main components. We have a phosphate group, a sugar, and a nitrogen-containing base. Here you can see a difference between RNA and DNA is in this 2' OH group of the sugar. So in RNA we have ribose, but in DNA we lack this hydroxyl group. So it is 2-deoxyribose, which makes it deoxyribonucleic acid. And then of course we have a variety of nitrogen-containing bases. So we have this class of bases called pyrimidines, which have a single ring. You can see we have uracil, cytosine, and thymine. Uracil is found mostly in RNA, and thymine is found only in DNA. Cytosine is found in both. And we also have another class of nitrogen bases which have two cyclic rings called purines, which have adenine and guanine, found in both DNA and RNA. And basically the sequence in which these bases appear is what determines the information encoded by that stretch of DNA. So as we discussed, this is what a nucleotide would look like. So a nucleotide is made up of the three units we saw earlier. We have a phosphate group, the sugar, and the nitrogen base. Because we have a thymine base attached to this nucleotide, it's called deoxythymidine monophosphate, or DTMP. And similarly, when we attach different nitrogen bases, we get different nucleotides. Okay, and the way that these nucleotides form a polymer is by forming this bond. Okay, so basically polymerizes here, a phosphodiester bond between the phosphate group and the sugar. So this phosphate group basically can bond with this hydroxyl group to form a phosphodiester bond. So here we have these linked nucleotides forming a DNA strand, and DNA molecule is actually made up of two DNA strands of opposite polarity, meaning that one DNA strand runs in this direction. Okay? The 5' end is the one where the phosphate is facing up. 3' end is the one where the OH is facing, right? Because it's a 3' hydroxyl. So one where the OH is facing is a 3' end. So this determines the polarity of the strand. So since this is the 5' end, this is the 3' end, we say this strand is running from 5' to 3'. And in the DNA molecule, it'll have a complementary strand which is running from 5' to 3' in the opposite direction, right? Because the OH is exposed here, phosphate group is exposed here. So we have two strands of opposite polarity. This sugar phosphate region is called the backbone. Right? Sugar phosphate backbone is on the exterior of the DNA molecule, and in between these two strands we have the nitrogen bases which form hydrogen bonds with each other. So basically this has formed a hydrogen bond with the corresponding base on the opposite strand. This is what is holding those two strands together. And these two strands basically twist to form the DNA double helix. You can see that the helix, the most common form of DNA, bDNA, has some very specific parameters. So that the distance between two adjacent base pairs is 0.34 nanometers. the distance for one turn in this helix is 3.4 nanometers. This is fixed for the most common form of DNA found in our body. There are other forms of DNA where these parameters vary, but that's outside the scope of this lesson. But the most important feature of DNA is that these two, the nitrogen bases can only bond with one other partner. That we can only form hydrogen bonds between thymine and adenine, cytosine and guanine. So essentially, if I have an adenine on this side, the only partner it can form a bond with is thymine. Similarly, if I have thymine on this side, it can only form a bond with adenine. Okay? And this basically, so you can see that if this sequence of bases on this side is known, then that forces the other side to adopt a complementary sequence. So this is the main feature of DNA which allows it to replicate effectively. Okay, that's why this is what informs the method of DNA replication that we have. Okay, and as I'm sure you're aware, adenine and thymine pairs form 2 hydrogen bonds, cytosine and guanine pairs form 3 hydrogen bonds, which means the bonding in this pair is slightly stronger than the bonding in this pair. Right? So you need a little more energy to break this interaction, which will be important as we go further into PCR. So as we discussed, basically the sequence of bases on one strand is informing the sequence of bases on the other strand. Right? It's forcing this to be complementary. So which means if I have only one strand of DNA, right, then I can start building up this other strand based on the sequence that I already know. And that is how we actually get our method of DNA replication, which is called semi-conservative replication. Which means that if I have this parental DNA, then each of these strands acts as a template, okay, to synthesize a new strand. Because this strand, right, so this blue strand was a parent strand. This orange strand is synthesized to be complementary to the blue strand. Okay, so this is now an exact copy of this. Similarly here, this is the opposite strand from the double helix, but this orange strand is again made to be complementary to it. So this orange strand will be identical to this original complementary blue strand. Right? So both of these DNA molecules will have the same sequence as the original parental DNA. Okay? So each one of these strands can act as a template for synthesis of a new DNA molecule. And that is how we can conserve the genetic information in our genome throughout the process of DNA replication. Okay? So these are other models of DNA replication that don't fit within our molecular biology framework. In the conservative model, these two strands would stay together and an entirely new strand would be synthesized. Whereas in the dispersive model, we would basically, this would basically break up and we would get our original parental DNA mixed with the newly synthesized DNA. But given the nature of the hydrogen bonding in our DNA, I think this is by far the most efficient method for DNA replication. And how we know that DNA replication is semi-conservative is by this experiment called the Messerschmitt-Stahl experiment. It's a very elegant experiment where basically we are going to use isotope labeling to distinguish between parental DNA and newly formed DNA. And we're going to analyze it using a technique we saw a few weeks ago called density gradient centrifugation. So essentially, to begin with, we have to label the parental DNA. So that is done by growing our cells in 15N, which is the heavy nitrogen isotope. Okay, so any DNA that is formed will incorporate this isotope of nitrogen. So that will be the nitrogen in the nitrogen bases. So all the parental DNA will be marked as 15N, and so when we run it on a density gradient centrifugation, this will be the sort of heavy DNA because that's the heavy isotope. So it'll the band will be slightly shifted compared to DNA, which is band will be slightly shifted compared to DNA, which contains a 14N isotope. Okay. So here you can see 14N containing DNA will have a band here. The parental DNA, which you've just labeled with 15N, will have a band here. And if we had a mixture of light and heavy DNA, we would get two bands, right, where the light and heavy would separate due to their different density. However, after we label the parental DNA, we then transfer the cells to a medium containing 14N, okay, for the time required for one generation. For E. coli, maybe about 20 to 30 minutes. We just grow it in this lighter isotope. Since DNA replication is semi-conservative, a new strand will be synthesized using the old one as a template. So each DNA molecule contains one parental strand, which is 15N labeled, and one newly synthesized strand, which is 14N labeled. So that means that this will essentially be a hybrid of the two parents. So when we extract this DNA and run it in a density gradient centrifugation, we don't get a pattern like this. Instead, we get a band which is midway between these two bands, right? Which means that the DNA that we have gotten is a mixture of 14N and 15N. So this would not be possible if we had a conservative model of replication, because our parental DNA would remain as is, so we should be getting a 15N band and a newly synthesized DNA. Right? So if this were the model, this would still be completely 15N, this would be completely 14N, so we should have gotten two bands. But that's not what we're getting, which indicates that it might be one of these two models, right? Where we are seeing a mixture of the parental and progeny DNA. So to now differentiate between these two models, we grow the cells for another generation and then extract the DNA. And then we see that while we're still getting the hybrid band, we're also getting a light band, meaning there is some DNA which is completely 14N, completely newly synthesized. Okay. If we were to look at dispersive DNA replication, then we can see that all of the newly formed DNA would be a mixture of 15N and 14N labeled molecules, right? So we would not be able to ever get a completely 14N labeled DNA. That is something we'd only get in the semi-conservative mode, with these molecules, okay, have used this newly formed DNA strand as a template, and as a result, both of these molecules will be completely light isotope because they're both newly synthesized, while these will remain the hybrid, which is exactly what we see. And we see that as we increase the generation number, the number of hybrids will remain the same, right? Because the number of original template molecules is remaining constant. But what is going to increase is the number of newly synthesized DNA, and so the 14N band will become more and more intense, while the hybrid band will become less intense in comparison. So this is how it was proved by Messos and Stahl that DNA replication is semi-conservative. Right. Since we now know that DNA replication is semi-conservative, we can then look at what actually happens in DNA replication. So this is an extremely complex process which requires a lot of players, and we look at them slightly as an overview to understand what factors we need to look at when we try to replicate this reaction in vitro. So to begin with, we all know that the DNA is a double helix, But if you want to use one of these strands as a template, right, it will be a bit difficult for a protein to travel along this double helix. So it would be better if we were able to separate these two strands, right? And then each of them can easily act as a template. So that begins by using this molecule called DNA helicase, which basically unwinds this helix. It's an ATP driven process which unwinds the two strands and frees them from the helix. Once the strand has been unwound, we don't want it to again go back and form a strand again, right? That is defeating the purpose of helicase. So for that we require a protein called single strand DNA binding protein, okay, which then binds to the single stranded DNA which has been freed by the helicase and basically keeps it in a single stranded form. So here you can see single stranded pre-replicative DNA without the single stranded binding protein is forming all sorts of secondary structures. You've gotten these hairpins and so on. And so when our replication machinery actually reaches this, it will be very hard for it to process this as a template. So what as the single stranded binding protein does is essentially keep this template nice and straight and keeps it in order for the replication machinery to act on. Okay, so new bases are basically added on top of the old bases by an enzyme called DNA polymerase. Okay. So there are a number of steps in DNA replication. Essentially takes place as follows. There are pre-marked initiation sites, sites for the initiation of DNA synthesis, DNA replication, called origins of replication. And once we have gone through this process and unwound two DNA strands, make sure we've gotten a nice single strand to act as our template. What happens is these initiation sites are recognized by DNA, another enzyme called DNA primase. And what DNA primase does is add short stretches of RNA at the initiation site. This needs to be done because the DNA polymerase cannot begin DNA synthesis de novo. Okay, it can only add nucleotides to a pre-existing polymer. So it requires some small region of RNA so that it can start adding DNA. Okay? So this will basically be elongated to form a new DNA strand. So the DNA primase will create this RNA primer at the initiation site and then dissociate. This RNA primer will then be recognized by our actual replicative enzyme, which is DNA polymerase. So in E. coli, the major polymerase is DNA polymerase 3. And what DNA polymerase 3 requires is a 3' hydroxyl group to which it can add a new phosphate group. Okay, so if we have this 3' hydroxyl group, what it's going to do is add the phosphate group here. Okay. To form the phosphodiester bond. So you can see that by definition, because of this property, DNA synthesis can only proceed in one direction, right? Because it has a requirement for this to be free. So if instead the DNA has a free phosphate group, it can't come and add a hydroxyl group to it. Okay? So as a result, the DNA synthesis can only proceed in one direction, which is 5' to 3'. Which means the template has to be in the reverse direction. So this has to be the 3' end of the template. Okay. And its complementary strand is always the one that is synthesized. which is in the 5' to 3' direction. Okay. So what DNA polymerase is going to do is essentially recognize this 3' free 3' OH, and then recognize the template base over here, and add the complementary nucleotide here and form the phosphodiester bond before moving on to the next base. And as it reads through the template, this is going to synthesize its complementary strand. Once it reaches this RNA primer over here, which is marking the next origin of replication, the DNA polymerase is going to dissociate, and we're going to get the activity of another enzyme called DNA polymerase 1. DNA polymerase 1 is a slightly different enzyme in that it has other activities too, in that it has something called 5' to 3' exonuclease activity, okay, which means it can remove nucleotides in a 5' to 3' manner. So what it is going to do is remove this RNA primer. Okay. It's going to remove this RNA primer and fill in this gap. So now this whole stretch, which had RNA primers in between, is going to be filled with DNA. Okay. And once the entire complementary strand is synthesized, we still have this regions, okay, between where the primer was and our original DNA strand. So these gaps, basically we need to make a new phosphodiester bond over here, and that is done by an enzyme called DNA ligase. Right? And through this whole procedure, we have basically synthesized one complementary strand of DNA. So just to give you an idea, this is the DNA polymerase is an extremely complex enzyme. You can see it's made up of a huge number of subunits. It's a very big complex. But yeah, this is E. coli DNA pol 3. This is the first DNA polymerase isolated. So this was the enzyme isolated by Arthur Condor. And this was the first basically DNA polymerase that was studied in the lab. Yeah, so just an overview of what the DNA synthesis process looks like. This is something called the replication fork. You can see because the strand is divergent like this, it does give the appearance of a fork. So here you can see an enzyme called topoisomerase, which we didn't discuss previously. And what topoisomerase is doing is acting to prevent supercoiling of DNA. So rather DNA, on top of its helical nature, has an additional supercoiling on top of that. And when we unwind the DNA double helix, okay, basically we are putting stress on the supercoiled region which is ahead of the replication fork. Okay, so the topoisomerase works to relieve that stress, okay, and make sure that the DNA doesn't get more and more twisted ahead of the replication fork. Here you can see this whole complex. This is replication complex, okay, called primosome, which consists of helicase, which remember is separating the two DNA strands. Right? It has primase, which is what is adding the RNA primer. Okay, we have single-stranded DNA binding protein, which is keeping these two strands separate. We have DNA polymerase 3, which is what is actually synthesizing our novel DNA strand. And we have ligase, which is going to fill in these gaps in the middle. Right? So you can see that in order to replicate DNA, essentially we have to go through through a number of steps. Right? First, we need to separate the strands. Okay. We have this double-stranded template. We need to separate the strands. Then we need to add a primer. Say this is one strand, and I want to synthesize a opposite strand. I need to add a primer here, right? So that the DNA polymerase can come and extend it. Okay, and once my primer is added, I need a DNA polymerase is going to come and add nucleotides according to the sequence of my template DNA. And this is essentially the basic principle of the polymerase chain reaction. Right? We just want to replicate the major DNA replication steps that we see in nature, and we're going to achieve that by a series of steps at different temperatures called thermal cycle. So yeah, just another look at the replication region. Since there are two strands, right, and as we discussed, a DNA double helix has two strands, and when we look at one will be 5' to 3', the other will be 3' to 5'. So this can act very effectively as a template, right? Because once, if the RNA primer sits here, it can just replicate like this. But if the RNA primer sits here, it cannot extend in this direction, right? This primer will be extended in this direction, because as we discussed, DNA is only synthesized in one direction, and it requires a template to go in the 3' to 5' direction. So how DNA is synthesized from this strand is by adding primers at different stages along the strand and extending them to each other. Okay, so basically forming a series of fragments which are then joined together. These are regions called Okazaki fragments. Okay, and this strand is called the lagging strand. So here you can see we've separated the DNA helix into this strand. This is the 3' to 5' strand, where it's easily replicated, and because this is going in the same direction as the direction of movement of the polymerase. So here we have replicating the leading strand continuously, as we discussed. However, on this other strand, right, we were adding the RNA primer, and this is now being replicated in the opposite direction. So DNA polymerase is adding it in the opposite direction to come to this previous RNA primer, where it will then be ligated by the ligase. So it's replicating the lagging strand discontinuously. Okay. But as we discussed, this unit is mainly about the polymerase chain reaction, which, as we discussed, is essentially replicating the DNA replication process in vitro in order to produce many copies of our target DNA. So we begin with our double-stranded DNA sequence of interest, and as we discussed, we require a primer and a DNA polymerase. Primer and a DNA polymerase, and we need to first separate these strands. Now, of course, if we had a helicase and single-stranded binding protein, they would separate the strands. But another way to separate or denature DNA is by heating it to 95 degrees Celsius. Heating it basically above 90 degrees Celsius, it causes these hydrogen bonds to break and separates these two strands. So just by heating it, we can break apart these two strands, and now we have both of these can act as templates. What we then need to do is anneal a primer, right? So the primer has to attach to a template. This is a process called annealing. And once the primer is attached, the DNA polymerase can see the free 3' OH of the primer, it can see the template, and it will start adding nucleotides according to the template. So this will be synthesized in this direction, while the strand will be synthesized in this direction. And we'll get, after the first cycle from one template double-stranded DNA, we'll get now two different double-stranded DNAs. And so you can see in each subsequent cycle, each of these can now act as a template. Right? So from 2 templates, we're now getting 4 molecules. And in this next cycle, we're going to get 8 molecules. So in each cycle, we're going to increase the number of DNA molecules by a factor of 2. So you can see that if we have a sufficient number of cycles, like say 20 or 30 cycles, we will actually be able to have millions of copies of our original DNA sequence. And that is the power of the polymerase chain reaction. So just looking at PCRs again, if we want to amplify a certain region within our genome of DNA, what we would have to do is design primers for the 2 ends of this region. So because the primer is defining the end of the region which is going to be amplified. So if we want to amplify this green region, we need to design primers that will bind to these 2 ends. Yeah, we have to design PCR primers to flank the region to be amplified. So one is going to bind to this strand, one is going to bind to this strand. So primers are complementary to the existing sequences. Right? And then this is going to be extended in this direction, this is going to be extended in this direction. And as the PCR process continues, you will eventually get PCR products that are contain this region between the 2 primers. So this is one PCR cycle where we have to separate the 2 2 strands in double-stranded DNA. So that is called denaturation. DNA denatured by heating to greater than 94 degrees Celsius. Then we need to anneal it. Okay. And in annealing, we are allowing our primers, which are basically oligonucleotide, to bind to the target sequence. And then we're going to allow DNA polymerase to extend it. Now, one thing you might see is that in this process, we are heating this DNA to a very high temperature, that's greater than 94 degrees Celsius. Most enzymes will not survive this kind of heating. Right? Even if the protein is still there, but it will probably not be active at this kind of temperature. So one option is that we start this cycle and then add the enzyme at this step, and then keep adding the enzyme at each cycle. But the process would be far more efficient if we had an enzyme that was able to retain activity even after heating to this temperature. And that enzyme precisely was found in an organism called Thermus aquaticus, which gave us a DNA polymerase called TAC, right? TAC for Thermus aquaticus, which is able to withstand this temperature and actually works best at 72 degrees Celsius. So this step you will actually be doing at 72 degrees Celsius because that's the optimal temperature for this enzyme. So you can see in the one PCR cycle consists of 3 steps: denaturation, annealing, and extension. And each cycle will increase the number of DNA molecules by a factor of 2. And as we repeatedly cycle through this process, we will produce exponentially more and more copies of our DNA. There are many forms of DNA that you can do. You don't have to use DNA as a template. You can also use RNA. So this is mRNA, right, which is poly A plus. So this is the poly A tail. And since we know that this has a poly A region, we can design a primer that is complementary to the poly A region. So we have a primer, a poly T primer, right? Poly T primer, and this is going to extend in this direction. Okay. You have to use a special enzyme in this case called reverse transcriptase, right? Because you're going from RNA to DNA, not from DNA to DNA, using an RNA template to form DNA. So we need a special enzyme called reverse transcriptase, which is going to form a new DNA strand, which we're going to call cDNA, okay, clonal DNA. And this cDNA can now be used as a template for PCR. Okay. So this is very useful when you want to look at the expression level of certain genes, right? Because that will be related to the amount of RNA that is present in the cell. So you can then quantitate the amount of DNA produced in the PCR, which will give you an idea of the amount of the original template. So, this process is called reverse transcriptase PCR or RT-PCR. RT-PCR is sometimes used as an abbreviation for real-time PCR, but that process is more technically called quantitative PCR or qPCR. And yeah, just an overview of what happens to DNA strands in the cycle. So we start with this template, anneal. This template gets extended all the way to the end, however much the DNA polymerase is able to add. This primer will get extended all the way to the limit of the DNA polymerase in this direction. But now when these two act as templates, right, so this is limited on this side by the primer. And this is limited on this side by the primer. So eventually the vast majority of the DNA products that we get will be capped by the primers on both ends and be of the size of the DNA enclosed by the two primers. Right? As the number of cycles in PCR increases, the DNA strands that are synthesized and become available as templates are delimited by the ends of the primers. Thus, specific amplification of the desired target sequence flanked by the primers is achieved. And just to give you an idea of the power of PCR, if we start with one template molecule, okay, then in the first cycle we don't have any newly synthesized molecules. In the second cycle, we're getting a hybrid of our template and our newly synthesized DNA. In the third cycle, we have two of our hybrid DNA molecules and two newly synthesized molecules. Right? In the fourth cycle, we'll have four, and so on, increasing in powers of 2. And you can see, as we get to the end of the 30th cycle, we have now synthesized an enormous amount of DNA. Right? More than a million, 268 million theoretically double-stranded target molecules have been synthesized through this exponential process. So what would a PCR reaction look like? So what components do you need in the PCR reaction? We need a template, we need primers, we need DNA polymerase, and we need deoxyribonucleotides, because those need to be added to form the new strand. So here we see we need input DNA, primer 1 and primer 2. You need to have two primers because that will define the two ends. Okay, that will amplify from both strands and define the two ends of your amplified region. These are buffer components that are required for the functioning of the polymerase. We need deoxynucleoside triphosphate. We need these because these are the building blocks of our newly synthesized strands, right? So we need a fair amount of these GNTPs in our reaction mixture. And of course, DNA polymerase. So what will happen is we will use a temperature program. So first of course for denaturation to separate the two strands. And then have to allow the primers to anneal. Right? If we keep it at 95 degrees Celsius, the primers cannot anneal because that is a denaturing temperature for DNA. So we need to lower the temperature to a temperature that allows the primers to bind to our template DNA strand. So the temperature at which we do this depends on the sequence of our primer. And so, but typically this is between 50 and 60 degrees Celsius. So we need to give some time for it to anneal, and then after that we take it to the optimum temperature for DNA polymerase and allow it to extend. Now, the length of DNA that you can amplify is determined by how good your DNA polymerase is. So, the standard PCR does not efficiently amplify sequences much longer than about 3 kb. Okay, because DNA polymerases have a certain quality called processability, which is basically the amount of DNA it can replicate before it then dissociates from the template. And for TAC, this will be around 3 kb. If you want to amplify longer sequences, you will have to look into using different DNA polymerases, and we discuss that later. So, one thing about TAC polymerase is that it does not have this a feature called 3' to 5' exonuclease activity. This is something called proofreading activity, because what this actually does is help the DNA polymerase recognize when it has incorporated a wrong base and get rid of it. So what the DNA polymerase is doing is synthesizing DNA in this 5' to 3' form. Right? So you're getting this new DNA strand in 5' to 3' direction. But what if we've accidentally incorporated the wrong base over here? So some DNA polymerases have the ability to recognize that this is a misincorporated base, get rid of it, and then reincorporate the correct base. So it can recognize that it has made a mistake and correct it, and this is called proofreading activity. Now this is not present in TAC. Okay, this TAC does not have proofreading activity, and so when you use TAC polymerase for PCR reactions, you will typically get a higher number of errors, right? Because it doesn't have a way of correcting those errors. However, there are some thermostable DNA polymerases, which means they can withstand 95 degrees Celsius, which do have proofreading activity. So here are some names: PMA, Liquent, PFU. So this is something called fusion polymerase, which is another widely used polymerase. PW. And you can see these are all from extremophile species, right? All species that live in these high temperature environments, which is why they have developed these enzymes in the first place. So we just discussed how we can use PCR to get many copies of our target DNA. But you can also see that what this effectively means is that the amount of DNA that we get, right, in a PCR cycle will be proportional to the number of target DNA molecules, right? Because here if I have one template, okay, then after one cycle I'll have 2, after 2 cycles I'll have 4, etc. But if I have 2 template molecules, then I already have 2 molecules in the first cycle. So after 1 cycle I will have 4. After 2 cycles I will have 8, and so on. You can see that the amount of amplified product that is created is proportional to the amount of original DNA that is present. And this is exploited in a technique called real-time PCR, or usually quantitative PCR, which looks at the amount of sample produced over time. This is normally used to quantify things like expression levels of different genes, compare and contrast to see what happens to the same gene in different conditions. So if we had a gene that was expressing very highly or overexpressed, then the number of number of product molecules that we get through this procedure would be much higher. And so in a lower number of cycles, we would start seeing an increase in the number of products. Whereas if our gene was being suppressed, okay, and expression had gone down, it would take a far higher number of cycles to reach the same level of amplification. Right? And so you can easily tell the difference between the relative expression level of these two. Let's discuss a little bit more about the essential components of a PCR reaction. So there are a couple of essential components. One is obviously a thermostable DNA polymerase to catalyze template-dependent synthesis of DNA. So a couple of things to consider when choosing a DNA polymerase. One is something called the fidelity, which is how accurately it is incorporating bases into your newly synthesized strands. So obviously this does incorporate the proofreading activity that we talked about earlier. If it has a proofreading activity, it will be more accurate, so it will have greater fidelity. Efficiency, this is how quickly it can synthesize your DNA, and ability to synthesize large DNA products. It's called processivity, which will limit the size of the PCR product you can generate. So for most standard PCR, stacked polymerase is fine. Right? If you want to so here you see efficiency of primary extension of stacked polymerase is generally about 0.7, which is telling you that if I incorporate 10 primers, TAC will be able to extend about 7 of them. So that gives you an idea of how efficiently it will catalyze our PCR reaction. There are enzymes which are far more efficient, have better fidelity, have better processivity than TAC, but they're usually more expensive, and so TAC polymerase is fine for most standard PCR reactions. We need a pair of synthetic oligonucleotides to prime the DNA synthesis. This is the primer. And what is very important is that these primers are designed well. Okay? If there are a couple of things to keep in mind when designing primers. One is that if you want to amplify a small region of this long DNA molecule, a primer should only bind in these two regions where they're supposed to bind. I should not have a primer that can bind here and also bind here. Okay, if this is the case, then I will get multiple products. If I have a primer that binds here and also binds over here, then I'm going to get one PCR product that is this big, one PCR product that is this big. Okay, and this is an unwanted product, but I'm getting it because my primer has multiple binding sites. So it's very important to design primers which are specific to the region that you want to amplify. Okay. It is also usually better if the two primers you use have a similar annealing temperature. Okay. The annealing temperature you use is dependent on the dependent on the sequence of the primer. Okay. It's basically dependent on the number of AT pairs and CG pairs that need to be formed. Okay. Since you already know that AT pairs will form 2 bonds and CG pairs will form 3. Therefore, the greater the number of cytosine and guanine nucleotides you have in your primer sequence, the higher the annealing temperature will be. So careful design of primers is required to obtain the desired products in high yield, suppress amplification of unwanted sequences, right? So we don't want our primers to bind in other places, and to facilitate subsequent manipulation of the amplified product. So if you do want to process your amplified product further, okay, say I have in this DNA sequence, I have this region here. What I want to do is clone it into a vector. Okay, I have a plasmid over here. I have a number of restriction sites here. What I want to do is clone this gene into this vector. Now what I can do is choose 2 restriction sites and digest this vector. So as a result, I get this empty vector which is ready for a gene to be inserted into it. But I'm amplifying this gene from genomic DNA. There's no restriction site that is going to be on both sides, conveniently going to be on both sides of my gene. So what you can actually do is add the restriction site as part of your primer. So this much, so you have a primer sequence which is complementary to the gene, right? Because you need that much to synthesize a PCR product. But beyond that, what you can do is add a small non-complementary region which can contain the restriction site. And so when we get our PCR product from this, it is now going to have restriction sites on both ends because we added it as part of our primer. Okay. And then we can digest this and ligate it into our vector. And so you can see that depending on what you want to do with your PCR product, you can essentially have to be very thoughtful about how you design your primers, very intentional. You have to keep in mind what your final end goal is. What else we need? We obviously need deoxynucleotide triphosphates or DNTPs. We should have equal molar amounts of the 4 DNTPs, because we will be using all 4 of them in reaction. Normally, we would need a concentration of 200 to 250 micromolar of DNTP. Now, usually we do use DNTPs in this range because high concentration of DNTPs can be inhibited. Okay, they can actually inhibit the PCR reaction. While this is ideal, we can also use some lower DNTP concentrations, but this is ideal and gives us the maximum amount of amplifier. Now, another thing you have to remember is, since we're using an enzyme, we do have to provide conditions which are ideal for the enzyme to function. And part of that in the buffer conditions is the use of divalent cations. So all thermostable DNA polymerases do require free divalent cations for functioning. This is usually magnesium 2+. Some polymerases can also work with manganese. Now, magnesium 2+ can be added externally, so we can just add a small amount of magnesium chloride to the reaction, and that will provide the Mg2+, which is required for thermostable DNA polymerases to work. One issue we do have is that DNTPs nucleotides can bind magnesium. So here you can see that if these do bind, then that is removing both the raw material for the template strand as well as the cofactor for the enzyme. Right? So it's obviously going to inhibit your PCR reaction if this interaction takes place between the DNTPs and the magnesium ions. So the molar concentration of the cation, that Mg2 plus that we use, must exceed the molar concentration of phosphate groups which is present in the reaction. Otherwise, this will not be available to the polymerase as a cofactor. So normally this will be about 1.5 to 2 millimolar magnesium 2 plus. And since magnesium 2 plus is required for PCR, we don't want chelating agents such as EDTA in the DNA that we're going to use. Sometimes when you're isolating DNA or isolating plasmids, you will resuspend the final DNA in tris EDTA buffer. Okay, that is to protect the DNA from nucleases. But if for PCR, it is better to avoid EDTA in the DNA resuspension buffer. So you can resuspend it instead in nuclease-free water. Okay, and that will basically make sure that we don't have any EDTA to chelate the magnesium which is required for our enzyme activity. So just an example of how important it is to use the optimal magnesium 2 plus concentration. Here you can see we using a range of magnesium concentrations from 0 to 6 millimolar, but our PCR product is only formed between 2 to 3.5 millimolar magnesium. So here the magnesium concentration is too low. That is all being sequestered by the DNTPs in the primer. It's not available to our nucleotide. And here it is too high. Basically, it has become inhibitory to the reaction. And this is essentially how you can analyze PCR reaction by running it on an agarose chain, as it is a DNA molecular. And the more DNA that is produced in the reaction, the brighter the band. Again, since we're using an enzyme, we want the enzyme to be happy, so we have to use a buffer that will keep it happy. We usually use a buffer containing tris-Cl, okay, and with pH between 8.3 and 8.8. Since we use tris is a temperature sensitive buffer. That means its pH changes drastically with change in temperature. So while it may be around pH 8.5 room temperature, when we go to 72 degrees, which is where the enzyme is actually active, the buffer will actually have a pH around 7.2. We sometimes, if you want to use, or rather, you can also include some monovalent cations, usually KCl, which is providing potassium, and 50 molar KCl helps in the amplification of long DNA fragments, which is more than 500 base pairs, whereas the slightly higher KCl concentration can help with the amplification of short DNA fragments. And of course, we need the template DNA, right, which is what is going to inform the sequence of the DNA which is produced in here. This can be either single or double-stranded DNA. Closed circular DNA templates can also be used, but obviously they will be amplified less efficiently because linear DNA will be more accessible than circular DNA. Now, the size of the template does not matter as such. If we have a very large amount of genomic DNA, okay, which can form multiple secondary structures, the amplification efficiency might be improved by first digesting the DNA, okay, and to make it basically a mixture of short templates, and then using this mixture as the template for a PCR reaction. Ideally, ideally, if you think about it, we require one copy of our template. However, we will normally use several thousands of copies of DNA, which can be up to 1 microgram of DNA per reaction. Normally, we can use about 1 nanogram of genomic DNA or 1 picogram of plasmid DNA as minimum for your PCR reaction. One thing to keep in mind when you're doing PCR is that it is very sensitive to contamination. So your template DNA will be amplified, yes, but if there is any form of contamination that can interfere with the enzyme, it can interfere with annealing and so on, it can cause a big effect on the nature of the reaction. So this is just looking at sterilized and unsterilized material. That is just, are you using autoclave tips or not? So right, so technically we're just adding mixture components. It's an enzymatic reaction. So you might think that it's not important to use things like sterilized tips or work in a hood and so on. But here you can see if we use unsterilized material, we get this smear over here. We're getting this over here. So, and this is our actual target. So this is our target amplified DNA. Okay, here you can see 225 base pair segment. We're getting a band around 200 base pair. This is our target. But we're getting all these smears of some DNA. Yeah, we don't know what it is, but this is some contaminating DNA. And here we're getting primer dimers. Okay, so this is basically the primer binding to itself. Okay. We have two primers that will bind on two ends of the DNA, but instead they are binding to themselves and forming this set of bands called a primer dimer. On the other hand, here you can see when they used a sterilized material, we get a much cleaner gel, right? Get a much better band as well. Band profile is much better, and we don't see any of the contaminating DNA that we saw in the previous gel. So it is important to use autoclave tubes, autoclave tips, use sterile water when you are performing PCR, because it is a very sensitive technique. So since we've learned a bit about PCR, we can talk a little bit, maybe go through some questions regarding PCR, such as, which is not a component of PCR workflow. So as we just discussed, magnesium is required for the activity of DNA polymerase. So just HCl is the buffering component, and KCl is often added as the monovalent cation to help amplification. So NaCl is a component which is not normally found in PCR workflow. Okay, what is the sequence for the PCR process? So one, denaturation of the double-stranded DNA. DNA polymerase binds the template primer hybrid, and dealing with the primers with sequences that clearly match template sequences. So this is clearly the first. Right? So first we have to denature double-stranded DNA. Then we have to anneal primers. Only after the primers have formed the primer template hybrid can the DNA polymerase bind. Right? So the correct sequence should be 1, 3, 2. Which of the following statements is true? PCR requires target DNA. This is true. 1 primer, all for deoxyribonucleotide and thermostable dependent polymers. So what we do know is PCR requires target DNA. Obviously, we need primers that are going to bind on both ends of the target DNA. So we need two primers. We need all four oligonucleotides because all four of them are present in our template DNA, right? By default. So we will need all four deoxyribonucleotides in a PCR mixture. And of course, we need a thermostable DNA polymerase which is going to incorporate them and synthesize and extract. So, yeah, we require target DNA, two primers, all four DNTPs, and thermostable DNA polymerase. Approximately how many pieces of sequence are needed for this DNA to be amplified for further analysis? So, two PCR cycles is going to increase your DNA amount by a power of 4. This is usually not going to be sufficient for any downstream processing that you want to do. So we will typically run a PCR of at least 20 cycles, which will give us at more than 1 million copies of the DNA, which will be sufficient for whatever downstream application you want to do. It is not necessary to perform this many cycles. Okay, Taq polymerase is isolated from bacteria, that is Thermus aquaticus is bacteria. Which of the following statements is accurate for the PCR polymerase chain reaction? Automated PCR machines are called thermocyclers. This is true because it is going through a thermal cycle. Right? We start then it is going to go down, then it is going to come up, and it is going to repeat this cycle. Normally, in a PCR reaction, this is going to repeat times. You will have an initial denaturation. So we have an initial denaturation where we will denature the template, go through the PCR cycle, have a final elongation step for any DNA molecules which haven't been synthesized fully. And then the product will be stored at 4 degrees Celsius. So since it is going through all these different temperatures, it's usually called a thermal cycle. Thermostable DNA polymerase is required, yes. Millions of desired DNA copies can be reproduced, so I think the correct option here is all of the above. Which is the following application of PCR? DNA cloning, medical diagnostics, for analytic analysis, and answer is all of the above. We will discuss a little bit about how we can use PCR in these. Primers used for PCR are single-stranded oligonucleotides. Now, while we use RNA primers in our body, what we use for is we can remove the RNA primer because we then have DNA polymerase 1 to remove the primer and synthesize new DNA stretch in its place. In PCR, we use a single polymerase, and for that reason, we use a single-stranded DNA oligonucleotide. Taq polymerase used for DNA amplification, and PCR is isolated from dash. Taq, it's there in the name itself. This comes from Thermus aquaticus. Which of the following is not a component of the PCR reaction mixture? DNTPs we need, DNA polymerase we need, primers we need, RNA polymerase we do not need because we are not synthesizing RNA. DNA polymerase used in PCR must have the following characteristics. Thermostability, of course. High fidelity, yes. We don't want it to incorporate erroneous bases. High processability, yes. So we require all of the above. How many types of DNTP is required for PCR? So we discussed we need all 4. Optimum GC content for primers and PCR must be in the range of usually 40 to 60%. Polymerase chain reaction was invented by a guy called Cadmus. You can go and read more. Nobel Prize winning. Which of the following is a thermostable polymerase? PFU and TAC are both thermostable. DNA pol 3 is not. So the option here is both A and B. The correct order of steps in PCR is dash. Obviously, first we need to denature, then we need to anneal, then we need to extend. So the correct option is D. A high temperature of 95 degrees is used in PCR for the purpose of dash. It is used for the purpose of denaturation, since we need to separate the two strands of the template. So we require it for separation of the new strands. We do not require for synthesis because our enzyme will not work at 95 degrees Celsius and our primers will not bind at 95 degrees Celsius. It's only used for separation of the template DNA strands. Okay, how many DNA duplex is obtained from one DNA duplex after 5 cycles of PCR? So what you need to know is after n cycles, so after one, if we start with one template, after one cycle we will get 2. So you can see after 2 cycles you will get 4. So here you can see if you have one template, we start with 2 raised to 0 molecules. After one cycle we will have 2 raised to 1. 2 cycles we will have 2 raised to 2. Right? So similarly after n cycles we will have 2 raised to n molecules of DNA. Right? So after 5 cycles we will have 2 raised to 5, which is equal to 32 duplexes, of which one will be our template DNA and the other 31 will be newly synthesized DNAs which will form through PCR cycle. DNA amplification due to PCR can be analyzed using agarose gel electrophoresis. We just run it on an agarose gel to check the size of the amplified product. It should match with our expected size of the region between the two primers. If it doesn't match, that means that there is some error in your primer design. Which of the following ions function as cofactor for DNA polymerases? We know that we have to supplement magnesium in the PCR reaction because it is providing a cofactor for the polymerase. Okay, which of the following statements are true regarding PCR? So, primer extension occurs at 72 degrees. This is true. Denaturation involves heating at 90 to 98 degrees Celsius. This is true. Annealing involves binding of the primer between 40 and 60 degrees Celsius. This is true. So, answer to this is all of the above. What would be the effect on the PCR reaction if any of the following circumstances occurs? 1. There are no primers in the reaction. 2. There are no DNTPs in the reaction. Or 3. There is no TAC polymerase in the reaction. So, as you can imagine, since we are taking out DNA components, so taking out components of the PCR reaction, we will not be able to amplify the DNA. If there are no primers in the reaction, we cannot start. Right? There's nothing for the DNA polymerase to add nucleotides to. There are no DNTPs in the reaction, there's no raw material for the synthesis. There's no TAC polymerase, there's no enzyme to actually do the synthesis. So the PCR reaction will not commence in this case. During the annealing step of PCR, which of the following steps occur? So, from a binds to target sequence, yes, this happens in annealing. DNA polymerase extends. However, no, this is the extension step. Template DNA stands separate. This happens in denaturation. And during annealing, the only thing that happens is the primer binds to target sequence. Okay. Extension temperature of 72 degrees of PCR is used for what purpose? So it is used because it is the optimum temperature for the action of DNA polymerases to extend the 3' end of the primer's complementary template. Okay, this was optimum temperature for thermostable DNA columns. So, since we discussed what PCR reaction is, we can also briefly discuss some applications of PCR reactions, and one is in the detection of things called molecular markers. These can be markers for diseases, markers for genotypes. It's basically any specific DNA sequence that you want to detect in our genome. Now, one way of detecting this on a genome level is by basically cutting the genome with different restriction enzymes, and then the nature of the bands that you get after this is going to be representative of the original sequence. So maybe from two different individuals you get a different band profile. This is something called restriction fragment length polymorphism. But the far more accessible and widely used techniques are all PCR based. And here you can see the number of PCR based techniques to detect different molecular markers. So one is called random amplified polymorphic DNA. Since you can see the word amplified, you probably understand that it is a PCR based method. And how this works is you have short random primers, some random 8 to 15 nucleotide sequence. Since it's short, it is probably going to bind multiple regions of your genome. Right? Since you've randomly generated it, you don't know where it will bind, but you do know it will bind in multiple regions. And we anneal it at low temperature, so we can allow it to bind to regions which are not exactly complementary. So if I have a region which is A, T, T, G, A, right? The exactly complementary primer would be T, A, C, T. And if I were to anneal this at say 55 or 60 degrees, this would be the only primer that could bind. But say I have a primer that looks like this. T, A, C, C, T. This is not exactly complementary to this, but it is mostly complementary. And this will be able to bind to this region of DNA even at a low temperature. Okay? Even though it is not exactly complementary regions will be able to bind at low temperature. So what happens in RAPD is that we have our randomly designed primers, and when we run the PCR reaction, we'll get randomly amplified products of various sizes. And now what is going to happen is, if we have some insertion or deletion in this region, okay, we have some insertion or maybe part of this deletion, then the size of this band is going to change, right? Maybe it's going to get longer, or maybe it might be shorter. And we're going to see that difference when we run the amplified fragments in a chip. So basically the length of the PCR product obtained is what is going to be characteristic of the marker that we're looking at. So this is what an RAPD gel might look like. Right? So you can see they're mostly similar except for this individual. It's 9th one. You can see that these bands, okay, are much fainter, and we have a new band over here instead, which means there might be some deletion or something in this individual. So if we have RAPD markers which are specific to say a gene for a disease, and we see a binding pattern like this, so this could indicate basically a positive result for the presence of this mutation. So RAPD can work with a small amount of DNA, obviously, since we're running PCR. It's a simple experimental setup, we're just running PCR. Okay? So which makes it widely accessible. But it can be difficult to interpret because we can have mismatches between the primer and template. It can result in both either no product or a decreased amount of product and so on. So we may get slightly conflicting results. And secondly, it is imperative that since we're doing a PCR reaction, the PCR cyclic conditions can greatly influence the outcome. So RAPD is usually a lab-to-lab dependent technique. It may not be reproducible as such in a different lab. A slightly better version of RAPD is something called amplified fragment length polymorphism, or AFLP, which is basically a combination of RFLP and PCR. So RFLP was just restriction fragment length polymorphism, where we are cutting up the DNA and then getting different restriction fragments. Amplified fragment length polymorphism is now going to amplify these fragments. Okay. Basically, you get these restriction fragments to digest with two restriction enzyme, get a series of fragments, amplify these fragments, and then detect them. And as a result, we get a complex and highly informative binding profile. Since we are going to amplify this by PCR, we need some region where our primer can bind. So we don't know what is the internal sequence of the DNA, but we do know what is the sequence of this end part, which is the region which is cut by the restriction enzyme. So what we can do is ligate a small stretch of DNA called an adapter, which is complementary to this cut site, and then design a primer which is complementary to the adapter. Right, so I have this primer complementary to the adapter. It's going to bind here and then extend in this direction. Design a primer complementary to this adapter. I can extend in this direction, and this way I can amplify this fragment even though I don't know the sequence of the fragment itself. Okay? Just because I know the sequences of these two adapters that we've put on both ends. And like we mentioned, as a result of AFLP, we get an extremely detailed profile, a lot of data, which can be very useful if you want to look at things like gene profiling, genome profiling, making things like phylogenetic analyses and so on. So it's extremely sensitive, high reproducibility, and since we're running a PCR, we can determine restriction fragments even if we have a lot of background. But it needs a fair amount of DNA since we have to run a restriction dilatation reaction. And they can be more expensive as they need more sensitive detection methods. So why would you want to look at molecular markers in the first place? So here you can see markers associated with resistance traits in crop plants. Okay, so these are basically resistance against various pathogens. So suppose we want to see if the rice that we are growing has resistance to this pathogen Xanthomonas oryzae. Then we can try and detect the presence of these genes, XA21, XA3, XA4, XA10, and these can be detected using these molecular marker techniques we just discussed. So this can be detected through RAPD. Using RAPD, a specific set of random primers, we will get a different profile depending on whether this gene is present or not. So instead of having to grow your rice plant, infect it and then see whether it is actually showing resistance, you can just run a PCR reaction and see if this resistance gene of your interest is present or not. Another kind of molecular marker is called a sequence specific repeat, an SSR, or a microsatellite. This is regions of DNA which are present in all eukaryotic genomes. basically was highly repetitive stretches, and the nucleotides that are present in this repeat are different in different species. So here in this comparison of plant species, we see that we can have repeats of these specific sequences. So in Arabidopsis, you have 80 repeats, right? So you have regions which just go 80, 80, 80, 80. What can vary between individuals is the number of repeats in this region. So here you can see we have two individuals who have this repetitive region, but this genome has 4 repeats, while this genome has 6 repeats between this flanking region. And so if we were to run a PCR reaction using these primers, this will give us a shorter PCR product, while this will give us a longer PCR product. So we can detect variations like this also using PCR. So yes, this is genotype with an 80 repeat. We know the flanking region of the SSR. When we run PCR, we see that this yields a longer product or this yields a shorter product, and we can use this information to determine the genotype of the organism. So an example of what an SSR detection gel will look like, you can quite easily see the difference in the size of the PCR product, which is the result of the different number of repeats present in the genome. So you can see that since we're running a PCR reaction, we can screen multiple markers at the same time. So it's a high throughput technique. It's highly reproducible, but we do need to know what the flanking regions of the microsatellite or highly repetitive region. So we can design primers for that specific region. So we do require sequence information about the genome in order to design these primers. So this is just a comparison table between various molecular marker detection techniques. I would say the most widely used is probably AFLP, because it is extremely sensitive and extremely reproducible. Another thing that you can actually detect through PCR is single nucleotide polymorphisms. So these are single nucleotide differences in the genome. Okay, so here you can see while the rest of the sequence is the same, at this one position, this individual has a C, this individual has a T, this individual has an E. So you can have single nucleotide polymorphisms. Change the codon, which is translated from this DNA, and even the single amino acid change can actually have a big effect on the protein product, because the protein which is formed to be misfunctional can cause it to be misfolded, can cause a significant change in the expression level, so on. So even SNPs can be markers for various diseases, and these can be detected through PCR. PCR technique called allele specific PCR, where basically you are looking at this depends on clever primer design, where you have a primer that matches allele 1, a primer that matches allele 2, and when you do perform PCR with these two and a common primer, you get 2 different bands, and that allows you to detect whether allele 1 or 2 is present, or even both. So you can see this lower band corresponds to allele 1, which has an A. This band over here corresponds to allele 2. And you can see these are both homozygous. This is homozygous for 1, this is homozygous for 2. This is a heterozygous individual because I'm getting bands for both. yeah, so that is how we can look at various applications of PCR. So this has yeah. What else? I wanted to discuss different. Also wanted to discuss some of the DNA polymerases that are available. What we already discussed is TAC DNA polymerase, which we use for routine PCR. And you can see in terms of fidelity, does it have 5' to 3' exonuclease activity? Yes. Etc. But it doesn't have a number of other features. But the main issue with TAC is that it is low fidelity and it will usually incorporate a fair number of erroneous nucleotides as you amplify your sequences. And so if we're looking at polymerases with high fidelity, you can see things like this polymerase called Q5, high fidelity DNA polymerase, which has fidelity 280 times of TAC. You will see fusion, which is 39 to 50 times TAC. So depending on the application, depending on what you want to do, there are a number of DNA polymerases available, which are all thermostable, all used for different kinds of PCR reactions. Another thing we could quickly discuss is how to design a primer. If I see, if I want to isolate a gene, I see a gene, I want to basically design primers that are going to amplify my gene on two sides. Okay, so if you pick up a random gene, you will see an option here called pick primers. So this is a primer picking tool within NCBI itself. And how big is it? 895 base pairs. So we want some primer in the initial region. We want some primer in the nature region so that we can isolate the full size. We want to amplify the whole genome, so the PCR product size should correspond to the whole gene. If you have some pre-designed primers, you can add them here, and you can give some guide to the primer melting temperature that you would like to keep. So this is telling you it's going to design primers with a melting temperature. This is basically the annealing. So this is the temperature at which your primer is going to dissociate from your template DNA. So annealing temperature will typically be slightly lower than your melting temperature, maybe usually Tm minus 3. So it's going to design primers with melting temperature between 57 and 63 degrees, and it's going to keep both the primers with the melting temperature in the same range. So it's a max Tm reference is 3. ask it to find primers for us. So while this is going to pick primers for a specific template, the other option is designing your own primers. And while we wait for this to design our primer, we can look at maybe designing our own primer. So say we have a gene, right? We have a gene with this sequence, and I want to design a primer to clone this gene. So what I need to do is design a forward primer at the start and a reverse primer at the end, which is going to clone the gene. So just manually, we want to look for a primer that is at least 20 base pairs long and will have a TM of under 65 degrees Celsius. So say I take this region, I want to design a forward primer. So here you see this is going to be my forward primer. It has 20 annealed bases, it has a TM of 63 degrees, and it has 60% GC content. So these are all pretty good conditions for the primer. Now, if you add this, suddenly I want to add some primer to the reverse region. But this one's reverse primer, so it's going to anneal to the bottom strand. So this is 18 bases, but one issue here is that this region is quite GC rich. Right? You can see 78% GC. So the TM for this primer is going to be relatively high, but that's okay. This has TM 67 degrees, which is still something we can work with. This is 60, TM 63 degrees, so this difference is still something we can work with in our PCR reaction. So if we have these two primers, what temperature should we now do our annealing at? Get this. Instead we look at what you can do is input the sequences of your two primers. And you can choose the polymerase that you're going to use. Okay? Say we're using a TAC polymerase, right? TAC polymerase with standard TAC buffer. Prime concentration. This will depend on how much you add to your reaction. Usually standard will be around 200 nanomolar. And here it is telling us the TM of these two primers as well as the optimal annealing temperature. You can see annealing temperature is lower than the melting temperature of these two primers. Remember, the melting temperature is the temperature at which the primer will dissociate from our template. You might see that depending on the polymerase we use, say want to use Q5, remember the polymerase with more higher fidelity. You'll see it tells us to higher in temperature high fidelity polymerase will differ from tactile polymerases. The buffer composition also favors higher TM values. So you do have to keep in mind which polymerase you're going to be using, as that can affect the annealing temperature that is ideal for your PCR reaction. So there are a number of tools online to basically look at your primer TM, tell you what is the ideal annealing temperature and so on. This is another thermos calculator. Where they will again tell you. Let's see. So earlier it was 69 degrees. This is also going to tell us. This calculator is telling us to anneal at 67.7 degrees. So all of this are just various resources that you can use to look at what the best way to use a primers is. So when we so one other thing you have to keep in mind is that when you design primers, they should not be binding to each other, right? Because that is going to defeat the purpose of your PCR reaction. They should be binding to your template, not to each other. So they should also ideally, since it is a single-stranded oligonucleotide, it can adopt different secondary structures. Ideally, you don't want your primer to adopt a high secondary structure. So you can use a tool like this, okay, which is going to look at your primer. So since I've input one sequence, it's analyzed it to tell me what its TM is, and it's looked at self-dimers, where it doesn't have any self-dimer. But if we input another sequence, it's also going to look at something called cross primer dimers, which is basically are our two primers going to bind to each other. And here you can see that our primers which we've designed are actually fairly good in that they are not binding to each other, right? Neither to themselves nor to each other. If instead of this I had a region like this in the primer, I had a primer that was basically a palindrome, then it is possible that they might bind to each other. So So you can see now, if I have a palindromic sequence, that there's a high chance that it can bind to each other. So it can form a self-dimer. So two dimers for sequence 1, where it is basically dimerizing with itself. So this, if I were to design a primer like this, it is more likely that it would form dimers with itself rather than binding to my template. So these are all things you do need to think about when designing primers, since they will directly affect the success of your PCR reaction. Okay. But yeah, there are many tools online to help you with designing your primers. I've just shown you a couple of them, but yeah, there are plenty of resources that you can make use of. As ever, if you have any questions related to the course, you can please feel free to use forum, discussion forum in the Google group. You can always make use of these problem solving sessions to ask me or any other course members about any doubts that you might have. And that's all I have for this week. So it's great